Welcome to the Deep Dive, the show where we journey through the cutting edge of research to extract the most vital insights just for you.
Glad to be here.
Today, we're plunging into one of medicine's most formidable and, well, enduring mysteries, cancer. And not just any cancer, but glioblastoma, an aggressive brain tumor that has stubbornly resisted our best efforts for decades.
It's a really tough one. While the broader field of cancer research has seen incredible progress, GBM remains.
Yeah, an exceptionally challenging adversary.
So our core topic for this deep dive is a groundbreaking study titled The Multilayered Transcriptional Architecture of Glioblastoma Ecosystems. Sounds complex.
It is, but the way they break it down is fascinating.
Imagine, if you will, that glioblastoma is a vast, complex city. Our mission today is to peel back its many layers like an urban planner studying every detail.
Right, from the individual residents to the bustling neighborhoods, the unseen infrastructure.
Exactly. All to truly understand how this incredibly intricate tumor city operates at its most fundamental level. And our mission for you, our dedicated learner, is to provide a clear shortcut to understanding precisely why the internal diversity of glioblastoma makes it so resistant to therapies.
Because that diversity is key, isn't it?
Absolutely central. This groundbreaking new study, using some truly innovative techniques, sheds brilliant light on this crucial area, giving us a far clearer picture than ever before. We're going to help you quickly, yet thoroughly, grasp the nuances of this complex and vital topic.
Okay, let's begin our exploration then. Let's unpack this. Glioblastoma, specifically the isocitrate dehydrogenase wild type, or IDH-WT-GBM, it's notorious.
That specific subtype, yeah. It's known for being incredibly tough to treat.
It often appears suddenly, grows relentlessly, and frequently returns even after aggressive treatment. But why? Why is it such a formidable adversary? What makes this particular brain tumor so stubbornly resistant?
Well, one core reason, as this new research powerfully illustrates, lies in its remarkable ability to diversify and adapt. This phenomenon is what scientists call heterogeneity.
Heterogeneity, okay.
So when we talk about IDH-WT-GBM, we're referring to a specific aggressive subtype. It's distinct from other gliomas that might have a different IDH gene mutation, which can sometimes mean a better prognosis.
Right.
This IDH wild-type version is actually one of the most common and malignant primary brain tumors in adults.
Yeah.
And its heterogeneity, its diversity, is a major part of the challenge. We essentially break this diversity down into two main types.
Two types. Okay, what are they?
First, there's intertumor heterogeneity.
Intertumor. So differences between different GBM samples, meaning the tumors from different patients?
Exactly.
Yeah.
So your tumor, my tumor, a neighbor's tumor, even follower glioblastoma, they each have their own unique characteristics at the genetic and transcriptional level.
Ah, okay. It's like that analogy you mentioned earlier, like every GBM patient's tumor being a unique snowflake, not just a generic one from a mold.
That's a perfect way to put it. They're all snowflakes, yes, all made of ice, fundamentally GBM, but each has its own intricate individual design.
Which must make developing a single one-size-fits-all treatment approach incredibly difficult.
Precisely. What works for one patient might not work for another, even if they seemingly have the same diagnosis on paper.
Okay, so that's between tumors. What's the other type?
And then, maybe even more challenging, we have intratumor heterogeneity.
Intra, meaning within a single GBM specimen, inside one tumor.
Right. Imagine taking a biopsy from just one patient's tumor. Even within that single sample, you'll find a dizzying array of cells.
Wow.
Some are malignant, meaning they're cancerous and actively growing. But there are also many different types of non-malignant cells coexisting: immune cells, blood vessel cells, even some normal brain cells trapped within.
So to stick with the snowflake analogy, even within that single unique snowflake, there are many different crystal structures and patterns coexisting and interacting.
Exactly. Each with its own characteristics and behaviors. It's like a miniature dynamic ecosystem within each tumor. Very chaotic one.
And this diversity isn't just a fascinating biological quirk, is it? It has profound implications for treatment.
Absolutely profound. This inherent variability, this constant shifting of identities and characteristics, it endows malignant GBM cells with distinct functional properties. That's a quote from the paper.
Distinct properties, meaning some cells might be good at invading, others at resisting drugs?
Precisely. It's exactly this adaptability that is thought to enable them to evade therapies, leading to progression and relapse. It's like trying to hit a moving, shape-shifting target. You might kill off one population of cells, but another resistant population survives and takes over.
And you mentioned the non-malignant cells, too.
Yeah.
The tumor microenvironment, the TME.
Yes, we can't forget the critical role of the TME. These non-cancerous cells form a complex ecosystem around and within the tumor, actively shaping GBM biology and its response to treatment. They're not just innocent bystanders, they're active participants.
Supporting the tumor, in a way.
Often, yes, they can provide growth signals, help build blood vessels, or even suppress the immune system from attacking the cancer cells. It's a whole complex interplay.
Right. Now, previous studies, especially using single-cell RNA sequencing or cRNA-seq, they have given us glimpses into this heterogeneity, haven't they?
Oh, absolutely. Invaluable glimpses. They were foundational. They showed that GBM malignant cells exhibit a variety of these cellular states, sometimes even resembling normal neurodevelopmental cell types. That was a key initial insight.
But as often happens in science, these initial explorations, while really important, they ran into some significant hurdles.
They did. It was like trying to see a detailed landscape through a slightly foggy window. You could make out the major features, the mountains and rivers, so to speak, but the finer details, the hidden valleys or the rare flowers, remained obscured.
So what were those hurdles specifically? This new study must have addressed them.
Indeed. The paper meticulously outlines 6 key limitations of those past srna-seq studies. First, they were often based on a limited number of samples and cells.
Just not enough data points.
Exactly. Studying cancer's immense complexity, especially finding those rare but potentially crucial cellular states, requires a truly massive scale. Previous efforts just weren't quite there yet. Imagine trying to understand a bustling city by only interviewing, say, 10 residents. You'd miss so much.
Makes sense. What else?
Second, a major technical hurdle was their reliance on enzymatic tumor dissociation.
Ah, the process of breaking up the tumor tissue to get single cells.
Right. To get individual cells for sequencing, researchers used, well, fairly harsh chemical enzymes. The problem is this process may deplete certain cell types and states.
Deplete them, like destroy them.
Or at least significantly reduce their numbers in the final sample. The paper notes this is evident by the scarcity of detected astrocytes, neurons, and endothelial cells in many older datasets. These are important players in the brain and the TME.
So the method itself introduced a bias, like that analogy you used, trying to extract delicate flowers from a garden, but your tools damage the most fragile ones.
That's a great way to think about it. Delicate cells with complex shapes, complex morphology, like neurons or certain glial cells, were likely destroyed or significantly altered by the enzymes. This leads to a skewed picture of what's really in the tumor.
Okay, that's a big one. What was third?
Third, and this is a crucial missing piece, past studies generally lacked an integrated model. They tended to look at either heterogeneity across tumors, comparing different tumor subtypes.
Like comparing different cities.
Right. Or they looked at heterogeneity within tumors, focusing on individual cell states.
Like looking at houses within one city.
Exactly. But they didn't really manage to develop a comprehensive model that explains tumor diversity by combining both tumor-level features, the city-level view, and cell-level features, the house-level view. We needed a map showing how the houses make up the city.
Gotcha. An integrated view was missing.
Precisely. Fourth, they often lacked in-depth genetic characterization of the tumors.
The underlying DNA changes.
Yes. Genetics are fundamental to cancer, driving many of its behaviors. Having detailed genetic information alongside the cell state data is critical for understanding the why behind the heterogeneity.
Okay. Fifth,
often limited clinical metadata, information about the patients, their treatments, how they responded.
Right, connecting the biology back to the patient experience.
Exactly. Without it, the biological insights lack crucial real-world context. And finally, sixth, these studies were often published by different groups, using diverse methods, different pipelines, analytics, classification schemes.
Making it hard to compare apples to apples.
Very hard. It made it difficult to combine findings, compare results reliably, and build a cohesive, broadly accepted picture of GBM's complexity. So the big question for any researcher then becomes, how do you overcome all these technical and logistical challenges to get a truly comprehensive, unbiased, and integrated view of GBM's complexity?
And that's precisely where this deep dive central study comes in. This is the solution they proposed.
Exactly. To tackle these monumental challenges, the researchers formed something truly special. The GBM Cellular Analysis of Resistance and Evolution, or CARE, consortium.
Right.
They essentially built a scientific super team, pooling resources and expertise from multiple institutions. and they developed a powerful new way to look at GBM.
Creating what they call a multi-omic data set of unprecedented size.
Unprecedented is the right word. It's like upgrading from that slightly foggy window we talked about to a high-definition satellite image combined with microscopic street-level detail, finally seeing the whole city in sharp focus.
So let's talk about the scope and methodology, the how behind this breakthrough. The scale sounds impressive.
It really is. They profiled 121 IDHWT GBM samples from 59 patients. And crucially, this included both primary tumors, the initial diagnosis, and recurrent tumors from the same patients after treatment.
Oh, wow. So they could see how the tumor changed over time and after therapy.
Precisely. That matched pair analysis is incredibly powerful. And 121 samples. That's a truly massive data set compared to previous efforts. It gives them the statistical power to uncover rare events, subtle patterns, and make much more robust conclusions.
Okay, massive scale. What about the technology? You mentioned innovative techniques.
Yes, the key technologies they employed were pivotal. First, they used single-nucleus RNA sequencing, or snRNA-seq.
Single nucleus, not single cell. What's the difference?
It's a crucial difference. Instead of trying to isolate fragile whole cells, which traditional enzymatic dissociation can damage, snRNA-seq isolates just the nuclei from the cells.
The cell's control center, basically.
Exactly, and it does this using a gentler process tissue homogenization in a cold buffer. This is paramount because, as the paper states, it eliminates the confounding effects of enzymatic tumor dissociation.
Ah, so it gets around that problem of damaging the delicate cells.
Precisely. It allowed them to capture a wider variety of cell types, including those typically underrepresented or missed entirely by standard cRNA-seq, like neurons and astrocytes. We actually see them clearly in their data figures.
So it's like, instead of trying to preserve the whole delicate flower, you just carefully remove the seed pod, the nucleus, the robust data chip inside.
That's a fantastic analogy. You get the internal blueprint, the RNA transcripts telling you what the cell was doing, without damaging the complex external structures that are sensitive to harsh processing. It preserves a much more accurate snapshot.
Okay, snRNA-seq is key. What else?
Second, they integrated this snRNA-seq data with bulk DNA sequencing, specifically whole exome sequencing, or WES, which looks at the protein-coding genes, and whole genome sequencing, or WGS, which sequences the entire DNA blueprint.
So they have the gene activity from the nuclei and the underlying genetic mutations from the DNA.
Exactly. This provided that in-depth genetic characterization that was often missing. It allows them to directly link specific genetic alterations, the DNA changes, with the single-nucleus transcriptomics data, which genes are active in which cells. That integration is incredibly powerful for understanding drivers of specific cell states.
And you mentioned clinical data, too.
Yes, and this shouldn't be underestimated. The data set is richly annotated with clinical data. They have a wealth of information about the patient's treatment histories, disease progression, outcomes.
Which is absolutely vital for making this clinically relevant, right? Connecting the molecular findings to what actually happens to patients.
Absolutely. It helps connect the dots from the lab bench to the patient's bedside.
So this comprehensive and integrated multi-omic approach scale, snRNA-seq, DNA sequencing, clinical data, this is what allowed them to make these breakthroughs.
Yes, it allowed them to achieve their key goals, identifying entirely new GBM cellular states, not just in the cancer cells, the malignant compartment, but also in the tumor microenvironment, the TME. And crucially, it allowed them to really start distinguishing the contributions of intratumor heterogeneity diversity within one tumor versus intertumor heterogeneity diversity between tumors.
It sounds like a massive leap forward.
It really represents a significant leap in understanding GBM's multilayered transcriptional architecture.
Yeah.
It gives us a much more structured way to think about this complexity.
Okay, let's do a quick recap before we dive into the findings.
Right.
So glioblastoma, specifically IDHWT GBM, is incredibly tough due to its heterogeneity both intertumor between patients, like unique snowflakes,
Right.
and intratumor within a single tumor, like diverse crystals in one snowflake.
Exactly.
Previous research hit limitations, small sample sizes, biased cell isolation from enzymatic methods that damaged fragile cells, and no integrated model combining tumor-level and cell-level diversity.
Correct on all points.
But this new study by the GBM Care Consortium used single-nucleus RNA sequencing, snRNA-seq, gently getting the data chip nucleus, plus bulk DNA sequencing on an unprecedented 121 samples, including matched pairs. This overcame past limits for a more complete, unbiased picture.
A perfect summary.
So what does this all mean for unlocking the secrets of GBM's hidden world? What did they actually find when they peeled back those layers with these new tools? Let's dive deeper into the discoveries, especially those brand new cell identities.
Okay. So remember those 4 principal cellular states in GBM that previous research had defined?
Yes, the MPC-like, OPC-like, AC-like, and MES-like, the sort of established categories.
Exactly. That classification scheme, which has been broadly adopted and validated, was our main framework. But this new, much larger data set allowed the researchers to significantly expand that atlas. They essentially almost tripled our view of the distinct malignant cellular states within GBM.
Tripled. Wow. How did they do that?
They used a powerful statistical technique called non-negative matrix factorization, or NMF.
NMF.
Okay, what does that do? Think of NMF as a sophisticated data sifting tool. It's an algorithm designed to find hidden patterns within complex gene expression data, like the data from thousands of single nuclei. Instead of just looking at individual genes, NMF helps drive underlying gene expression programs, groups of genes that tend to be activated together and vary across cells within each tumor.
So it's finding recurring themes or patterns in the gene activity.
Precisely. Like identifying recurring musical motifs in a complex symphony. Each motif, or program, represents a potential distinct cellular state or process. After running NMF and carefully filtering out programs that might just be technical noise, they found 10 groups of similar programs recurring across the dataset. They then defined a consensus metaprogram, or MP, for each of these 10 groups.
The 10 MPs. So these represent the distinct malignant cellular states in their massive dataset.
That's right, 10 distinct malignant metaprograms.
And how do these relate to the original 4 states?
Well, what's remarkable is how these new findings both confirm and refine our existing understanding. Overall, 7 of these 10 MPs largely recapitulate the patterns from previous studies, but they do so with greater resolution, finer detail.
Okay, so the familiar ones are still there.
Yes, the AC-like, astrocyte-like, OPC-like, oligodendrocyte progenitor-like, MPC-like, neural progenitor-like, and MES-like, mesenchymal-like states are clearly represented among these 10 MPs.
That provides validation then.
It does, but there are key refinements. For instance, cell cycle genes involved in cell division was identified as its own distinct MP. It wasn't just seen as a feature of other states, it is a state or process itself.
Okay, so active division is its own category. What else was refined?
And here's where it gets really interesting, particularly regarding the old mesenchymal-like category, which was known to be quite broad. This study elegantly split what was often lumped together. Hypoxia, related to low oxygen conditions and previously sometimes denoted as MES2, was identified as a separate MP.
So the response to low oxygen is its own distinct state.
Yes. And also stress, which includes genes associated with heat shock, and the unfolded protein response, basically cellular stress responses, was also identified as a separate MP. This was previously often just included within the broader MES-like definition.
So the old MES-like state is now split into 3. MES-like proper, hypoxia, and stress.
Exactly. What's fascinating here is how the sheer scale and the high-resolution analysis allowed them to do this. It highlights the nuanced complexity. Even within what we thought was one broad category, there are distinct biological responses happening.
It's like discovering that a single type of tree, in our city analogy, maybe one we just called hardwood, is actually 3 different species: oak, maple, and birch.
That's a perfect analogy. Oak, maple, birch, MES-like, hypoxia, stress. Each might have distinct needs, unique responses to environmental stressors like drought or pollution, and therefore potentially different vulnerabilities we could target. This granularity is crucial.
Okay, that refinement is huge, but you said they tripled the number of states, so there must be completely new ones, too.
Yes. And this is where it gets really fascinating for you, the learner, looking for those aha moments. This study identified 3 entirely new malignant cellular states identities that were largely missed or couldn't be clearly defined in previous glioma studies.
3 completely new ones. Hidden faces of glioblastoma.
You could definitely think of them that way. Revealed now thanks to the improved methods and scale.
Okay, tell me about them. What's the first one?
The first new state they found is called NEU-like, which stands for neuronal-like.
NEU-like, so resembling neurons.
Yes. This metaprogram contains some genes that were initially seen in progenitor-like states, but it was mostly associated with markers of more differentiated neurons. We're talking specific genes involved in neuronal development like NRG1 and NRXN3, and even synaptic activity like SYN3 and SYT1.
Differentiated, so more mature, less like stem cells.
Exactly. Consistent with that, cycling cells, those actively dividing, were actually depleted in this NEU-like program compared to the more progenitor-like MPC cells. They're not rapidly dividing.
Interesting. Do they resemble any specific normal brain cells?
They do. The analysis showed the highest similarity to specific types of excitatory neurons, like L2, L3 excitatory neurons, found during normal brain development.
And why was this missed before? Was it the dissociation problem again?
Very likely, the paper states. This state is depleted in single cell relative to single nucleus data, suggesting that it may be poorly captured by enzymatic tumor dissociation, possibly because of complex cellular morphology.
Ah, those delicate neuron-like structures getting damaged by the enzymes.
Exactly. So the snRNA-seq, by using the nucleus, that robust data chip, allowed them to finally see these fragile neuronal-like cells within the tumor consistently.
Okay, NEU-like. What's the second new one?
The second new malignant state they identified is GPC-like, which stands for glial progenitor cell-like.
GPC-like, glial progenitors. So related to stem cells.
That seems to be the key feature. This metaprogram included significant genes like the tyrosine kinases, EGFR and ALK, along with several transcription factors like MIS1 and ETD1. What's really significant is that these genes have been previously linked to glioma stemness or cancer stem cell properties.
Stemness, meaning self-renewal driving tumor growth.
Precisely. This strongly suggests that GPC-like state plays a crucial role in the tumor's engine of growth and its ability to regenerate. It showed the strongest association with a recently described signature of normal glial progenitor cells found in the healthy brain.
And are these cells dividing?
Yes, consistent with a progenitor or stem-like state, the GPC-like MP was highly enriched with cycling cells. These are actively dividing and self-renewing cells.
And presumably missed before for similar reasons?
Exactly the same reason, the paper notes. Like the NEU-like state, GPC-like cells were depleted in single-cell relative to single-nucleus data. Again, it underscores the superior capture method of nRNA-seq for these potentially delicate, complex progenitor cell types. This is a really key discovery for understanding how the tumor sustains itself.
Okay, NEU-like and GPC-like, two major finds. What's the third new state?
The third new state is truly unique, cilia-like.
Cilia-like, like the little hair-like structures on some cells?
Exactly those. This metaprogram mostly contained genes related to cilia function and structure, including many members of the DNAH and CFEL gene families. Cilia play crucial roles in signaling and sensing the environment in normal cells.
Is this state common?
Actually, no. It was quite rare, specific to only a small fraction of cells, about 1.6% of the total malignant cells they analyzed.
Wow, that's small.
Yes, which really underscores the utility of having such a massive cohort in high-resolution techniques. You need that scale to reliably identify these truly rare, yet potentially significant cellular states that might be involved in communication or sensing the microenvironment.
Is it related to any other state?
Interestingly, yes. They found that all of those cells expressing the cilia-like MP also express the AC-like MP.
Ah, so it's a specific subset of the astrocyte-like cells that activate the cilia program.
Precisely. It's like a specialized function adopted by some AC-like cells.
These three new states are fascinating. NEU-like, GPC-like, cilia-like.
And these findings collectively really support the idea that GBM states often mirror developmental trajectories observed in a healthy brain. It seems the cancer cells hijack or mimic normal developmental programs.
And the ability to identify these rare and delicate cell types, NEU-like and GPC-like especially, is a direct result of the snRNA-seq.
Absolutely. It's a direct result of the meticulous methodology, avoiding damaging these cells that possess complex processes, similar to normal neurons or progenitor cells.
Yeah.
It's like finally having the right tools to find those elusive species hidden deep in the jungle.
So these new and refined cell states, all 10 of them now, they aren't just labels, right? They must have different functions, different behaviors within the tumor.
Exactly. They come with specific functional attributes, meaning they might behave differently, which is crucial for thinking about potential therapeutic strategies. Knowing how a cell acts can tell you how to target it or what role it plays.
So what did they find about their functions?
Well, using computational analyses, they found some interesting patterns. For instance, AC-like and MES-like cells showed greater connectivity.
Connectivity, like forming networks?
Potentially, yes, suggesting they might be involved in forming structural networks within the tumor, or perhaps communicating extensively with each other, maintaining the tumor's architecture.
Okay, what about the others?
In contrast, OPC-like, MPC-like, and the new NEU-like cells had greater motility and invasion scores.
Motility and invasion, so they're the movers, the ones spreading through the brain.
That's strongly suggested by the gene programs they express. They seem geared towards movement and invading surrounding tissue, which is a key feature of GBM's aggressiveness.
And the other new ones, DPC-like and cilia-like.
They were described as intermediate in these connectivity and motility scores, suggesting maybe a different primary role.
And how confident are they in these new states? Were they validated?
Yes, importantly, these new metaprograms were rigorously validated. They weren't just artifacts of this one data set or analysis method. They detected them in data generated using a different sequencing platform called SmartSeq2.
Okay, different technology, same result. Good.
They were still present when they analyzed only the primary untreated samples, meaning they aren't just something that emerges after recurrence.
Also important.
And they were independently detected in specimens processed at different labs and medical centers within the consortium.
So robust findings across different conditions.
Very robust. This validation really confirms the reliability and generality of these new classifications.
Okay, let's recap this section too, because that was a lot of new information. So the study used that NMF algorithm on their massive data set to expand our view of GBM malignant states from the previous 4 to now 10 distinct metaprograms, or MPs.
Correct. A major expansion of the cellular atlas.
They identified 3 entirely new states missed before. NEU-like, resembling differentiated neurons, GPC-like glial progenitor cells linked to stemness and proliferation, and a rare cilia-like state, a specialized subset of AC-like cells.
Those are the 3 big discoveries, yes.
And the cRNA-seq method, preserving those delicate cell nuclei, was absolutely crucial for finding NEU-like and GPC-like cells, which enzymatic methods likely destroyed or missed.
Crucial methodological advantage.
And finally, these 10 malignant states aren't just categories. They have distinct functional characteristics like connectivity, ACMEs-like, versus motility and invasion, OPC, MPC, NE-like, which told us about their roles in the tumor.
Exactly. Functional implications are key.
It feels like we're really gaining a much richer understanding of the individual cell personalities within our GBM city analogy. But what happens when these personalities interact? Or when we zoom out to see the larger patterns of the entire tumor? Let's explore the dynamic shifts between cell states and those overarching baseline differences between tumors next.
Okay, moving from the individual cells to their dynamics in the bigger picture.
So imagine the cells in a GBM tumor aren't static entities, right?
Yeah.
Not fixed in their roles. But maybe they're constantly shifting their identities, like actors changing roles in a play or those shapeshifters we mentioned earlier.
That's a very apt analogy. The study found compelling evidence for exactly that, what they call hybrid states. This gives us a really dynamic, almost cinematic look at GBM evolution in action.
Hybrid states, meaning cells that look like they're in between states.
Precisely. The researchers found that roughly 20% of the cells could be classified into multiple states. What that means is, based on their gene expression, these cells scored significantly for at least 2 different malignant metaprograms, MPs, with only a limited difference between their top 2 scores. They didn't fit neatly into just one box.
20% is quite a lot.
That's-
Could that just be a technical glitch, like 2 cells stuck together?
That's a critical question they addressed. They meticulously checked if these could be technical doublets. 2 nuclei accidentally getting sequenced together in the same droplet. But hybrid cells did not have a higher number of detected genes, which you would expect from doublets.
Ah, okay. So it's not just a technical artifact, it's real biology.
That's the conclusion. Thus, we reason that these may represent cells in transition between 2 states.
Like seeing a caterpillar halfway through becoming a butterfly. It's neither fully one nor the other, but actively changing.
Exactly. It's a snapshot of cellular plasticity in action. And based on which states were most often found together in these hybrid cells, they constructed what they call the GBM cellular hierarchy model.
A hierarchy model,
Right.
showing how cells might transition between states.
Kind of like a potential roadmap. They found that certain pairs of states were highly enriched as hybrids, suggesting these are common or preferred transition routes. This model provides a fascinating glimpse into GBM's potential developmental hierarchy, showing us how cells might move between different identities. This could be key to the tumor's adaptability.
So what did this hierarchy model look like? Were some states more central than others?
Yes, there was a clear structure. They found that the more differentiated cellular states like NEU-like, AC-like, cilia-like, MES-like, and hypoxia were all positioned at the edges of this model.
the endpoints of differentiation, maybe? Cells that have specialized?
That's likely the interpretation, yes.
Oh.
In contrast, the three progenitor-like and stem-like states, NPC-like, OPC-like, and crucially, the newly discovered GPC-like state, were all found in the center of the model.
The center, like origins or hubs for transitions.
Exactly. Highly plastic populations from which other states might emerge. And specifically, the GPC-like state, that new progenitor one, was positioned right at the center of three major trajectories in their model. One leading towards NEU-like cells, another towards AC cilia-like cells, and a third towards hypoxia MES-like cells.
Wow. So the GPC-like state seems like a really key central hub.
It really does. The positioning perhaps suggests that this GPC-like population represents a truly early progenitor state with remarkable differentiation potential along multiple different paths. It could be a major source of the tumor's diversity and adaptability.
Now, can they tell the direction? Like, does a GPC become a NEU-like, or can a NEU-like go back to being GPC?
That's an important caveat. This analysis, based on snapshot data, lacks evidence for strict directionality. We don't know for sure if a cell always moves from A to B, or if B can revert to A, or if it's even more complex. However, even without definitive direction, the model offers a powerful story or hypothesis about tumor evolution and how cells might adapt.
And it fits with the idea of cancer hijacking normal development.
It really does. It reinforces the view that many aspects of GBM biology might be inherited from the original cell that became cancerous and can mirror processes seen in normal brain development. It underscores the tumor's dangerous plasticity.
Okay, that's the dynamic shifting within a tumor. Now let's zoom out again to the whole tumor level, looking between different patients' tumors.
Right, moving from intratumor dynamics to intertumor differences.
Beyond just the mix of cell types, those MPs, the researchers wanted to find other consistent patterns of differences between tumors. What makes my GBM fundamentally different from yours at a whole tumor level?
This is a critical question. And they developed a very clever and robust method to get at this, something they call a cell state controlled approach.
Cell state controlled, how does that work?
It's a significant innovation, another powerful algorithm in their analysis toolkit, designed to separate different layers of heterogeneity. It works in a few steps. First, they took each tumor sample and computationally broke it down into subpopulations of cells representing 7 common malignant states, the main MPs we've discussed.
Okay, so sorting the cells by state within each tumor.
Yes. Then second, for each subpopulation that had enough cells, at least 25, they calculated a state-specific pseudo-bulk expression profile.
Pseudobulk, like an average.
Exactly. It's like getting the average gene expression snapshot for, say, all the AC-like cells in this specific tumor, or all the MES-like cells in that specific tumor, getting the overall vibe of one neighborhood in the city.
Okay, an average profile for each state in each tumor. What next?
Third, and this is a really clever part, they then compared these pseudobulk profiles across different tumors, but only for the same cell state.
Ah, so comparing apples to apples, like comparing the average AC-like profile from tumor 1 directly to the average AC-like profile from tumor 2 and tumor 3 and so on.
Precisely. This is key because, as they state, it eliminated state-specific signals because they're only looking at one state at a time and highlighted tumor-specific differences in that state. By doing this comparison, they could identify patterns of gene expression variation that were inherent to the whole tumor, consistently showing up across multiple cell states within that tumor, independent of the specific mix of states.
I see. Isolating the tumor's underlying signature that affects all cell types within it.
That's a good way to put it.
Yeah.
Finally, they used principal component analysis, or PCA, a statistical method for simplifying complex data and finding the main axes of variation on these cross-tumor pseudobulk comparisons. This allowed them to define high and low gene signatures for the top 3 principal components, the biggest sources of variation.
And what did this reveal? Did they find consistent tumor-level pattern?
They absolutely did. This whole approach led to the identification of 3 sets of signatures, which they called state-controlled baseline profiles, or BPs.
Baseline profiles, BPs.
These BPs reflect fundamental patterns of intertumor heterogeneity differences between tumors that commonly influence several malignant states within a tumor. These are patterns that define the entire tumor's character, not just one cell type within it.
So distinct from the MPs, the cell states themselves.
Yes, this is a key insight. They found 3 major BPs, which they annotated based on the functions of the genes involved.
Yeah.
NEU, neuronal, glial, and extracellular matrix, ECM.
NEU, glial, and ECM BPs.
Think of the BPs as the overall atmosphere or maybe the background music of the entire tumor city, defining its fundamental character. In contrast, the malignant metaprograms, the MPs, are like the individual songs or distinct neighborhoods within that city.
Atmosphere versus songs. I like that. They're related but different levels.
Exactly. And critically, BPs reflect consistent malignant gene expression differences between tumors that are not accounted for by differences in tumor cell state distribution, the mix of MPs. They represent tumor-level phenotypes distinct from the internal shifts in cell states. MPs, remember, preferentially varied within tumors. BPs vary between tumors.
Okay, this distinction is important. Are these BPs consistent within a single tumor?
Remarkably so. First, they found that within a given tumor, the BP classification tended to be consistent across different cellular states in the tumor.
Meaning,
For instance, if a tumor was classified as having a high ECM BP, then not just the MES-like cells, but also the AC-like cells and the OPC-like cells within that same tumor tended to express higher levels of the genes defining the ECM BP.
wow, so it's like a pervasive flavor throughout that specific tumor, affecting multiple cell types.
That's a great analogy. It permeates the entire tumor, regardless of the specific cell state.
What about across different regions of the same tumor? Is the atmosphere the same everywhere?
Good question. To check that, they leveraged a GBM spatial transcriptomics dataset. Spatial transcriptomics lets you see gene expression patterns while keeping the location information where in the tissue the expression occurred.
So they could compare different neighborhoods in the same city?
Exactly. And they found that sample pairs taken from different areas of the same tumor had significantly higher similarity in their BP scores than samples taken from different tumors.
Meaning the overall BP atmosphere is largely uniform throughout a single tumor's mass.
That's what the data suggests, yes.
And do these new BPs relate to older classifications like the TCGA subtypes?
They do. They found a significant association between these newly defined BPs and the Cancer Genome Atlas TCGA expression subtypes, which are well-established classifications based on bulk tumor RNA sequencing. Specifically, they noted the greatest overlap between the BP-ECM and the MES-TCGA subtype.
So it connects to and potentially refines existing knowledge.
Absolutely. Taken together, these three baseline profiles, NEU, glial, ECM, reflect core tumor-level transcriptional variation. It's the fundamental phenotype of the tumor itself, partially independent of the variation in cellular states within that tumor. Gives us a crucial new lens, moving beyond just cell types to the whole tumor's underlying identity.
Okay, let's recap the section on dynamics and baseline profiles. So the study identified hybrid state cells scoring for multiple MPs as compelling evidence of dynamic cellular transitions within the tumor. Cells aren't fixed, they're shifting identities.
Like shapeshifters, yes, showing plasticity.
And among these, progenitor-like states, especially the newly discovered GPC-like state, appear to be central hubs in a hierarchy model, potentially differentiating into various other malignant states.
A key insight into the tumor's adaptability engine.
Then, zooming out, they introduce baseline profiles, BPs, NEU, glial, and ECM. These represent consistent tumor-level differences between tumors, distinct from the cell state MPs within tumors. Think atmosphere versus songs.
A crucial distinction, highlighting a new layer of intertumor heterogeneity.
And these BPs are consistent within different cellular states of the same tumor, like a pervasive flavor, and correlate with established TCGA subtypes.
Right. Validated and context-rich.
It's truly a sophisticated dance happening both inside individual tumors and across different patients. But the full picture isn't complete without considering the neighborhood, the surrounding environment of the tumor, the TME, and maybe the puppet master genetics pulling some strings. Let's explore those interactions next.
Absolutely. The ecosystem isn't just the cancer cells. We need to look at the whole environment and the genetic drivers.
Right. No tumor exists in isolation, does it? It's constantly interacting with its surroundings, its cellular neighbors, forming this complex ecosystem that can profoundly influence its behavior.
Constantly. It's a dynamic interplay.
So the researchers asked, how does the diversity within this tumor microenvironment, the TME, influence those malignant cell states, MPs, and the overall baseline profiles, BPs, we just talked about? Does the neighborhood shape the residents and vice versa?
To investigate this, they applied their NMF framework, that pattern-finding algorithm, not just to the malignant cells, but also to derive TME metaprograms for each non-malignant cell type.
Okay, so finding patterns in the immune cells, blood vessel cells, etc.
Exactly. Oligodendrocytes, astrocytes, neurons, endothelial cells, and importantly, TAMS tumor-associated macrophages, which are key immune cells often co-opted by the tumor. This resulted in an impressive 62 total MPs just for the TME.
62! Wow, that's incredible detail for the non-cancerous part.
It really is. It shows the complexity of the microenvironment itself. And their findings showed significant associations between the TME and the cancer cells.
What did they find?
First, TME-MP abundances differed significantly across GBMs, mapping to the 3 BPs.
Meaning the overall tumor atmosphere, BP, is linked to the specific types of non-malignant cells present.
Strongly suggests that, yes, the overall tumor phenotype is tied to its microenvironment composition. It's like the climate of our city impacting the types of non-cancerous support structures or inhabitants that thrive there.
And did the TME link to the specific malignant cell states, the MPs?
Yes. Similarly, TME-MP abundances were also associated with malignant MP abundances, indicating direct crosstalk at the cellular state level between malignant and non-malignant cells.
Can you give some examples, specific interactions they found?
Sure. For instance, they found the abundance of MES-like malignant cells was associated with increased tumor-associated myeloid cells, TAMs, and monocyte-derived macrophages, MDMs, but also with endothelial cells.
So the aggressive mesenchymal state seems to hang out with specific immune cells and blood vessel cells, maybe recruiting them.
That's a likely interpretation. It highlights how a more aggressive tumor state might actively recruit or be supported by certain immune and vascular components, potentially fueling its growth and spread.
Any other examples?
What's particularly fascinating, and a newer observation enabled by siRNA-seq capturing neurons, is that the abundance of NEU-like malignant cells was associated with neurons.
The cancer cells resembling neurons associated with actual non-cancerous neurons.
Yes. This suggests that non-malignant neurons, the healthy brain cells surviving near or within the tumor, may not just be bystanders. They might actively interact with these NEU-like malignant cells and shape their states, or vice versa.
That's a critical observation of crosstalk between the tumor and the normal brain tissue it's invading.
It really is. Additionally, the abundance of the newly discovered GPC-like cells was positively correlated with TAM microglia, a specific type of immune cell found in the brain. And the abundance of OPC-like malignant cells correlated with non-malignant OPCs.
So like attracts like in some cases.
It seems that way sometimes. Overall, it tells us the malignant cells aren't just evolving on their own, they're this constant dynamic dance with their environment. Specific TME cell types influence or are influenced by specific malignant cell states.
You mentioned hypoxia earlier. How does that fit in?
That connection was strong too. Malignant hypoxia, stress, and MES-like states were intimately associated with specific TME states. Things like inflammatory and hypoxic macrophages, reactive astrocytes, which are astrocytes responding to injury or inflammation, and inflammatory and angiogenic endothelial states related to blood vessel formation.
So a hypoxic stress tumor core might promote an inflammatory vessel-building environment around it.
That's the picture it paints. And this suggests a critical implication. Hypoxia in the TME might not just starve the tumor cells directly, but could also actively cultivate an immunosuppressive ecosystem around them. This environment could limit the effectiveness of treatments like immunotherapy that rely on the patient's immune system attacking the cancer.
Wow. So the TME isn't just passive scenery, it's actively shaping the battleground.
Absolutely. It's a key player.
Now, can they get even more specific? Beyond just associations, can they see how these cells might be communicating?
Yes, they took it a step further. To get more granular on how these cells might be talking to each other, they used a computational tool called CellChat.
CellChat, what does that do?
It quantitatively infers specific ligand receptor interactions between different cell populations based on their gene expression.
Ligand receptor, like a key fitting into a lock.
Yeah.
Specific molecular messages.
Exactly. Ligands are the signaling molecules, the keys, and receptors are the proteins on receiving cells that bind them, the locks. CellChat predicts which cells are sending which signals and which cells are receiving them. Think of it like decoding the actual secret messages these cells send to each other using specific protein words.
Decoding the tumor's secret communications network, what did they find?
Well, the analysis revealed two separate subnetworks of communication, which seem to align with the idea of distinct regions within the tumor, maybe tumor core, versus the periphery, each with its own unique communication patterns.
And specific messages. Any examples with functional implications?
Yes, they highlighted several key predicted interactions. First, they observed IGF-1, IGF-1R crosstalk. IGF-1 is the ligand, IGF-1R is the receptor. This was predicted between TAM-MDMs, those immune cells, and the MES-like malignant cells.
And why is that significant?
Because this specific interaction pathway is consistent with a proposed resistance mechanism. It's thought to promote cell survival and growth, so this communication might be actively helping the tumor resist therapy.
Okay, a potential resistance pathway decoded. What else?
Second, they identified PTN-PTPRZ1 OPC crosstalk. This interaction between different cell types expressing these molecules may promote tumor progression and invasion. So, another conversation potentially driving aggressiveness.
A target for stopping spread, maybe?
Potentially, yes. And third, remember the NEU-like cells associating with neurons. CellChat found predicted NLGN, NRXN, and NRG-ERBB4 interactions between neurons and NEU-like cells.
The specific molecular conversations between them.
Exactly. And significantly, these particular interaction pathways have already been reported as potential therapeutic target in glioma in other research.
So this confirms potential targets at the molecular communication level.
It does. These specific molecular dialogues really highlight how different TME niches, whether it's hypoxia, glial cells, or neurons, actively serve as determinants, shaping both the malignant and non-malignant states. Understanding these precise dialogues, these conversations, opens up powerful new avenues for targeted intervention. Maybe we can disrupt the tumor's communication network.
Okay, so the environment, the TME, plays a huge role, and they're starting to decode the specific messages. But what about the tumor's own inherent blueprint, its genetics? Do specific genetic alterations push the tumor towards certain states or BPs? Is there a genetic master switch?
That's the other critical piece of the puzzle, connecting the genetics to the transcriptional states. The study first confirmed several associations that were consistent with prior research.
Like what?
For example, they found that CDK4 amplification, a common genetic change, was associated with having more NPC-like malignant cells. PDGFRA amplification was linked to OPC-like abundance, and NF1 mutations were associated with MES-like abundance.
Okay, so established links between specific genetic drivers and certain cell identities were confirmed. That builds confidence.
It does. It shows their data set aligns with known biology. But interestingly, they also noted something that didn't align with some previous assumptions. They did not observe an association between EGFR amplifications and AC-like abundance.
Oh, EGFR amplification is a very common GBM alteration, right? And it was thought to be linked to the AC-like state.
It often was, based on broader studies. But in this detailed single-nucleus analysis, that specific link wasn't apparent, suggesting the relationship might be more complex or indirect than previously thought. Science is always refining its understanding.
Okay. But were there any new major genetic associations found, especially with those new cell states?
Yes. And here's a really significant new insight that ties back directly to one of our newly discovered states. The strongest associations they found were between the most common defining GBM genetic changes, what researchers call the canonical alterations, and the increased abundance of the GPC-like state.
The GPC-like state, that new glial progenitor stem-like state, linked to the core GBM genetics.
Exactly. This is a huge connection. When we talk about canonical genetic alterations of GBM, we mean the big ones. Often chromosome 7 gain, chromosome 10p loss, EGFR amplification, and TERT promoter mutations. These are the hallmarks of classic GBM.
And these often occur together, right?
Yes. They typically co-occur in GBM, forming a powerful genetic signature. And the study found a striking difference based on this signature. Tumors with these canonical alterations had, on average, 12% GPC-like malignant cells.
Okay, 12%. What about tumors without those core genetic changes?
They had less than 1% GPC-like cells, on average.
Wow, that's a massive difference. 12% versus less than 1%.
It's a statistically significant difference. Wilcox and Ransom test p equals 0.003, meaning it's highly unlikely to be due to chance. This suggests a really powerful link. The GPC-like state, that newly identified progenitor population, is rare in GBMs that lack these common major genetic changes.
So it implies that these core genetic drivers, like gaining chromosome 7 or amplifying EGFR, might actually predispose a tumor to developing this specific, highly proliferative GPC-like population, driving its aggressive growth.
That's a very strong implication from the data, yes. It suggests these canonical alterations might converge on activating or expanding this GPC-like progenitor state.
Any other interesting genetic links?
A few others were noted. TP53 mutations seemed linked to NEU-like abundance. And chromosome 7 amplification, besides being part of the canonical set, was also linked specifically to hypoxia abundance, which aligns with some recent studies suggesting genes on chromosome 7 help cells adapt to low oxygen.
And how did genetics relate to those overall baseline profiles, the BPs?
They found that the glial BP is more closely aligned with canonical GBM genetic alterations compared to ECM or NEU BPs.
So the tumors with the glial atmosphere are the ones most likely to have those classic GBM genetic changes, like EGFR amplification.
That's what the association suggests. It all starts to tie together. The canonical genetics, the glial BP, and the GPC-like state seem to form a related cluster of features. These results clearly indicate a complex interplay between specific genetic alterations, the resulting cellular states, and the overall tumor phenotype. It adds another critical layer to our understanding.
It really is like solving a giant multi-layered puzzle, isn't it? Let's recap this section quickly. The tumor microenvironment, TME, is not passive. Its diverse cell population significantly influence both malignant cell states, MPs, and the overarching baseline profiles, BPs.
Active crosstalk, definitely.
Ligand receptor interactions, those specific molecular conversations decoded using cell chat, reveal precise communication pathways like IGF-1, IGF-1R that shape tumor behavior and resistance.
Potential targets for disrupting communication.
And genetic alterations are deeply connected. Canonical GBM genetic changes like EGFR amplification, CHER7 gain-10 loss are strongly linked to the abundance of the newly identified GPC-like progenitor state and also align with the glial BP.
Genetics, cell states, and tumor phenotype are all intertwined.
This incredibly detailed analysis shows us GBM isn't just rogue cells, it's a dynamic interacting system, a true ecosystem. Which leads us finally to the study's ultimate breakthrough, the concept of integrated GBM ecosystems. What do these look like and what do they mean for the future?
Right. This is where they pull all these different layers together into a cohesive picture.
So we've walked through these three distinct, crucial layers of GBM transcriptional patterns. First, the overall cell type composition, how much malignant versus non-malignant tissue, and which TME cells dominate.
The basic cellular makeup.
Second, the specific malignant cellular states present those 10 MPs, like NEU-like, GPC-like, MES-like, defining the personalities of the cancer cells.
The malignant cell identities.
And third, those overarching baseline gene expression programs, the BPs, NEU, glial, ECM, defining the tumor's unique atmosphere.
The tumor-level phenotype.
The big question the researchers tackled was, how do these three layers interact? Do composition, malignant states, and BPs tend to associate in predictable, recurring patterns? Do certain neighborhoods and atmospheres consistently cluster together in our GBM city?
That was the goal, to see if integrated recurring patterns emerge from this complexity. To do this, they first systematically classified all 121 samples according to each of these three layers.
How did they classify them?
Okay, for cell type composition, tumors were first grouped by their malignant fraction, basically. The percentage of cancer cells into high, intermediate, or low. For those with a low malignant fraction, meaning lots of TME cells, they were further subdivided by the dominant TME cell type. Was it TAM-rich? Lots of macrophages. Oligodendrocyte-rich, glioneural, GN-rich, more normal brain cells like neurons, astrocytes, were just mixed.
Okay, classifying by overall composition.
Then, for the malignant state layer, they looked at the dominant MP in each tumor. Most tumors had one state that was clearly most abundant. This allowed them to define 6 groups: AC dominant, mesh epoxia dominant, GPC dominant, OPC and PC dominant, and UU dominant, or a mixed category if no single state clearly dominated.
Classifying by the main cancer cell type present.
Exactly. And finally, for the baseline profiles, BPs, tumors were classified into the 3 groups we already discussed: BP-NEU, BP-ECM, or BP-glial, based simply on which BP had the maximal score in that tumor.
Okay, so every tumor sample now has labels for its composition, its dominant malignant state, and its baseline profile.
Right. This systematic classification across all 3 layers allowed them to then look for statistical associations. Do certain labels consistently appear together?
And what do they find? Did patterns emerge?
They absolutely did. This was the groundbreaking part. When they looked for statistical associations across these 3 layers, specific features consistently clustered together. It wasn't random. This allowed them to define 3 multi-layered ecosystems.
Multi-layered ecosystems, wow.
Each ecosystem includes correlated features among all 3 layers: composition, malignant state, and BP. It's like finding 3 distinct recurring species, or archetypes of GBM tumors, each with its own characteristic makeup, behavior, and likely underlying genetic drivers.
3 main archetypes emerging from all that complexity. That's huge. Can you describe these 3 ecosystems?
Yes, let's delve into each of these 3 stereotypic GBM ecosystems, which they illustrate really well in the paper, Figure 6AB.
Okay, ecosystem number 1.
First, we have the BPNEU ecosystem. This one is characterized overall as glioneural enriched.
Okay, BPNEU, glioneural enriched. What are its features?
Within this ecosystem, the dominant malignant states are associated with the NEU-like and OPC-NPC-like MPs. So these tumors have more of those neuron-like or progenitor-like cancer cells. The TME composition is distinguished by a glioneural G-enriched TME, meaning a higher presence of non-malignant neurons, astrocytes, and oligodendrocytes. It's like a tumor growing within or interacting heavily with a more intact neuronal environment. And genetically, this ecosystem showed links to specific mutations in TP53 and RB1, which are well-known tumor suppressor genes.
Okay, so NEU-PCNPC states a glioneural TME and TT53RB1 mutations tend to cluster together. That's ecosystem 1. What's the second?
Second, there's the BPECM ecosystem. This one is characterized as mesenchymal-like enriched.
BPECM mesenchymal-like enriched. Features.
Here, the dominant malignant states are strongly associated with the MES-like and hypoxia MPs we discussed. So these tumors are driven by those mesenchymal and low oxygen response features. The TME in this ecosystem is typically macrophage-rich, meaning a higher presence of those TAM immune cells. And genetically, this ecosystem linked to NF1 mutations, another common GBM alteration often associated with mesenchymal features. This paints a picture of a more inflammatory, hypoxic, potentially invasive tumor.
Right. MS hypoxia states macrophage-rich TME, NF1 mutations clustering together, ecosystem 2, and the third.
And third, the BP glial ecosystem. This one is characterized as glial progenitor-like enriched.
BP glial, GPC-like enriched. Tell me more.
This ecosystem is strongly associated with that newly discovered GPC-like state, really emphasizing its importance. These tumors have a high abundance of those highly proliferative glial progenitor-like cancer cells. The TME in these tumors is often characterized by a highly malignant fraction, meaning fewer non-malignant TME cells overall. The tumor is densely packed with cancer cells. And genetically, this ecosystem is strongly linked to those canonical GBM drivers, EGFR and MDM2 amplifications.
Oh, connecting back to the GPC-like states link with canonical genetics.
Exactly. It all comes together. This BP glial ecosystem, dominated by GPC-like cells, with a high malignant fraction and driven by classic GBM alterations like EGFR amplification, really paints a picture of a very aggressive, highly proliferative tumor archetype.
This integrated model, it feels like the closest we've come to seeing GBM holistically.
It really is. It powerfully suggests that often these features, genetics, TME composition, malignant states, overall BP, don't exist in isolation. They cluster together in predictable ways. For example, if a tumor has EGFR amplification, it's significantly more likely to also have lots of GPC-like cells and fit into that overarching glial baseline profile ecosystem.
Which provides a potentially much more powerful and actionable framework for classifying GBM, right? Moving beyond just single genes or single cell types.
Absolutely. It gives us a framework for thinking about distinct biological entities within what we call glioblastoma. However, it's important to add a note of caution, reflecting the study's honesty about the complexity. While 43%, 52 of 121 of the samples, could be robustly assigned to one of these 3 multi-layer ecosystems.
Only 43%, so just under half fit neatly into one of these 3 boxes.
Right. The remaining samples reflect unique combinations of features. They didn't fit perfectly into one stereotype. This highlights that even with this massive breakthrough in finding these patterns, the diversity and complexity of GBM remains a significant challenge. Not all tumors are easily stereotyped.
It's an incredible stride finding these 3 major archetypes, but the journey to understand the full spectrum continues.
Precisely. It's a huge step, providing a new map, but there are still territories on that map that are unique or less well-defined.
So let's pull it all together. What are the big takeaways from this deep dive for you, our curious learner? What's the so what from all this groundbreaking work?
Well, first and foremost, this study provides a comprehensively redefined GBM cellular architecture. The scale and methodology, especially using sRNA-seq, was absolutely critical. It enabled the discovery of those 3 new cellular states, NEU-like, GPC-like, and cilia-like, which were previously hidden due to technical limits. This fundamentally expands our parts list for GBM.
A better atlas of the GBM city.
Exactly. Second, it really highlights the incredible cellular plasticity of GBM. Finding those hybrid states gives us direct evidence of cellular transitions, reinforcing the dynamic, shape-shifting nature of GBM cells and how they might mimic normal developmental processes to adapt.
Understanding that plasticity is key to understanding resistance.
Absolutely. Third, this research introduced a truly new layer of heterogeneity, the baseline profiles, BPs. These define a novel layer of intertumor heterogeneity, explaining more the variation between tumors in a way that's distinct from, yet correlated with, the cell states within tumors. It adds another dimension to our understanding of why GBM is so diverse across patients.
The atmosphere layer, complementing the cell state layer.
Right. And finally, the study culminates in that integrated model, the 3 stereotypic ecosystems. The overarching speculation or hypothesis from the researchers is that tumor genetics, along with tumor location, which influences the TME, define the propensity of each GBM toward a particular BP and enriched MP, culminating in 3 stereotypic ecosystems.
So genetics and location influence the TME, which shapes the BPs and MPs, leading to these ecosystems. A cohesive story.
It provides a cohesive potential story for GBM development and progression, connecting the tumor's intrinsic genetic blueprint with its microenvironment and its resulting cellular characteristics into a single comprehensive framework.
And the ultimate goal, of course, is treatment.
Right. How does this help patients?
Understanding these multi-layered ecosystems, how tumor genetics, the non-malignant TME, and the 2 malignant features, BP and MP frequency, all intertwine, is absolutely critical for developing more effective, personalized therapies for glioblastoma. It gives us a much more sophisticated map to guide treatment development.
This deep dive has truly given us an unparalleled, highly detailed view into the complex, dynamic world of glioblastoma. We've moved far beyond just individual cells to understanding entire integrated tumor ecosystems.
It's a huge conceptual shift. It really feels like a massive step forward in the ongoing fight against this challenging disease, offering new avenues for research and, hopefully down the line, more effective treatments, maybe even treatments that target an entire ecosystem, not just one component.
That's certainly a hope for the future. The revelation of these stereotypic ecosystems, while we have to acknowledge the remaining complexity that still needs study, provides a powerful new framework. We can think about GBM not just as one disease, but perhaps as a collection of distinct, interconnected biological entities that require more tailored approaches.
It really changes how we map that GBM city.
It absolutely does.
So thinking ahead, what does this new integrated understanding of GBM mean for future treatment strategies? If these ecosystems are, to some extent, stereotypic, could we develop therapies that target not just one cell type or one genetic mutation, but maybe disrupt an entire ecosystem within a tumor, attack its communication, its structure, its key cell types all at once?
That's the exciting long-term vision this kind of research enables, moving towards ecosystem-targeted therapy.
And for you, our dedicated learner, digesting all this complexity, how might this powerful framework of multilayered ecosystems change the way you approach and think about other complex biological systems or diseases? It feels like a template for understanding complexity itself.
It's a valuable way to think about many complex systems, definitely.
Lots to ponder. Until next time, keep digging deep.